Let's hear it from Dr. Newman. Welcome.
Hello, thank you. Okay, good. Okay, and so this system is one of the earliest, actually beginning in the 1970s, that was approached by a mathematical analysis. Let's see, the slides are not advancing. Sorry. Okay. Approached by a mathematical analysis based on physical processes. And in this case, the reaction diffusion mechanism that was pioneered by Alan Turing that I spoke about in my talk yesterday. And when the Turing idea for the limb was first proposed, it was actually confronting an older view that was characteristic of the way development was looked at in the 1960s and 70s, and it was more of a cybernetic approach. So let's consider these two approaches. So this is a limb bud from a vertebrate organism like a mouse or like a human. And we have to try to understand how the fingers, for example, will form within this tissue, because the tissue is quite homogeneous looking to begin with. And the earlier approach, the more cybernetic approach, said that every cell is like a little computer program. Its genome is a program. It has a representation of all the parts of the body in it. And all that's needed is some kind of a gradient shown by this shaded region here. And the cells read the gradient and they make fingers of different types, digits. And these are because there is a kind of internal representation in the cells, and all they need is this positional information in the tissue, and they just read the positional information and they make the different digits. This is the outcome, a stained preparation. The more physically oriented approach that was proposed in the late 70s said that there's a physical process that creates these chemical waves, these non-uniform peaks and valleys of some molecule. And here you see all the fingers when they first emerge are the same as each other. They may be slightly different sizes, but it's the same thing because you have a wave-like process that just activates the tissue in different places. And then later on, these can become kind of customized, like cars coming off an assembly line that are all identical to begin with, but then they become customized. So the key difference here is that here the cells are reacting on the basis of some internal program, but here you have interacting signals that organize the tissue in this repeated fashion, and that the differences only appear later on, and then you get the same outcome. So this is the kind of physico-genetic or generic approach, and this is a more programmatic cybernetic approach. And I'm going to show you that this is the view that actually has prevailed and is widely accepted now. But it took a long time for it to set in because this idea was very popular for many systems. And actually systems like segmentation and gastrulation gave way to the more physical approach way before the limb did. The limb was kind of the last to succumb to this physicogenetic approach. Okay, so just some definitions to begin with. This is a chicken limb during development, and it's shown transparent, and you can see the bones become established close to the body first, and then as time goes on, different stages, you get the entire skeleton laid out. And the entire skeleton is cartilage first. So the process in this tissue is called chondrogenesis, the development of cartilage. And it happens in what's called a proximal distal order. Proximal is the part close to the body, and distal is furthest from the body. And here, these axes are shown on a human hand, just more familiar to us. So proximal is the part that's close to the attachment point of the body, and distal is out to the fingertips. Anterior is the thumb end of the limb, and posterior is the little finger end. So there's a antero-posterior axis, and dorsal is the back of the hand, and ventral is the front. So there are three axes that are kind of landmarks for understanding development. And when we go back here, we can see there is a proximal distal progression of the laying out of the elements. Okay. Now, an important thing that happens before cartilage even differentiates is that the tissue inside the limb, it's called mesenchymal tissue, and you have these cells that are scattered and they're embedded in a jelly-like material called the extracellular matrix. And when the cartilage region is going to form, I'll go back and show you, when the cartilage first begins to form, it begins as this light gray trace here, and that is a condensation. These are condensations, meaning that the cells in this tissue move together and clump. And that is the first sign that cartilage will form there. And we can see that that happens progressively over time. And it happens because there is some additional material shown in this brown shading here that appears just in those sites where you're going to get condensations forming. When the condensations form, the cells signal to each other and they differentiate into cartilage. Later on, in most species, the cartilage gets replaced by bone. So the first patterning process is this condensation process. Okay, now one of the ways that we study these mechanisms, particularly using chicken or mouse tissues, is that we isolate from a limb bud, like a 5-day limb bud, we isolate a piece of tissue that has not yet formed any cartilage or even formed any condensations. We disperse the cells and we put them in culture. And this is about 3 millimeters across here. And the dimensions of the limb are, this is a total of about 7 millimeters in diameter. So this culture represents, it's a blown up version of this, but the culture forms many little nodules. And these nodules are condensed cells. So the cells between them are also mesenchyme. They're not seen here because they're not forming these dense nodules. But anyway, we study this nodule formation in culture, and it shows patterns that are on the same spatial scale as the patterns that we see in the limb. Actually, this is much smaller than this. This is really less than a millimeter here. So each of these little digits is about the same scale as one of those nodules. This is really about a half a millimeter. Okay, so the basis of the Turing type of mechanism, this physical mechanism of patterning, is that there are activator and inhibitor interactions. This is one of the forms that the Turing mechanism can take. So you have cells here, and the cells are producing an activator. And the activator is a autocatalytic or positively autoregulatory activator. So when the cells produce it, it reflects back on the cells and causes it to produce even more. And then the cells condense in response to this activator. And what happens is that eventually, because it's an explosive autoactivation, the whole tissue mass will organize into a big nodule of cartilage. But if at the same time the activator induces the production of an inhibitory molecule, and the inhibitory molecule diffuses away faster than the activator does, then you get this repeating pattern where you have centers of activation, condensation, chondrogenesis separated by spaces that are inhibited from undergoing this process. So when you have these two components, this was the inside of Turing, when you have these two components, you wind up with an inherently pattern-forming mechanism. They have to be balanced in an appropriate way for you to get the pattern, but once they're in balance, you get this kind of pattern. Okay, so is there any evidence for anything like this? Well, we looked at this using probes against proteins that are called galectins. And this work was done by Ramanujan, who's here at the meeting when he was a graduate student with me. And just about everything I'm going to be speaking about from now on was participated in and led in many ways by Ramanujan with some other colleagues of ours. What Ramanujan found was that there were a set of galectins that had been found in the chicken genome, and the chicken genome specifies 5 different galectins, and 2 of them play a key role in this Turing type of process. And so the galectins can be separated into a number of categories, and they're called galectins. You may be familiar with the term lectins, which are plant proteins that bind to carbohydrates. And these are basically animal lectins. They're endogenous lectins of animal tissues, and they also bind carbohydrate ligands. And the ligands that they bind on the cell surface always end in a galactoside, a beta-galactoside. So they have binding sites for the beta-galactoside, shown as these scooped out areas here. And they have a number of different formats. And in mammals, there are a dozen or more galectins, and they're all enumerated over here. Primates have more than rodents, but there's a whole bunch of them. Birds have very few galectins. They have the fewest among all of the vertebrate classes. And the ones that are in boxes are bird galectins, chicken galectins. There's galectin 1, galectin 2, galectin 3, and galectin 8. And they have different arrangements. This is 2 different proteins that are attached to each other. Here's one protein that's attached to a non-galectin tail, and here are 2 galectins that form a dimer, a non-covalently attached dimer. But in fact, in birds and in reptiles, the sauropsids, galectin 1 is duplicated. And this is not the case in mammals. It's the case in fish and amphibians. You have several galectin 1s, but in mammals, you only have 1 galectin 1. In sauropsids, you have 2 galectin 1s. They're called galectin 1A and galectin 1B. And what we found was that if we looked at the development of the limb, either in situ, in the actual limb bud, or if we looked at the progress of chondrogenesis in culture, galectin 1A was highly expressed just at the time that condensations were forming. And here is RNA staining of the RNA for the galectins in the condensation region. And it happens just at the time that condensation is taking place. And similarly with galectin 8, there is an upregulation during condensation. And here we're looking at 2 days, 3 days, 6 days during chondrogenesis in culture. And this is a lower magnification than this, but you see these nodular sites. And the galectin 1A and the galectin 8 coincide in their staining, and they stain at both the RNA and protein levels. They stain the condensations. Galectin 1B, 2, and 3 are expressed to much lower or negligible extents during this period. Okay, now doing a whole bunch of experiments that involved knocking down these galectins with inhibitory RNAs, or overexpressing the galectins by injecting messenger RNA, or using other methods of blocking or enhancing, we found that the phenotype that we got, and this is a stained limb, and you see the skeletal structures. This is a normal limb. This is the humerus, the radius ulna. Here are the digits. This is the control. If we add galectin 8 to the tip of the limb, we're missing structures. But if we knock down galectin 8, we get extra structures, ectopic structures that are not usually part of the limb. Similarly, if we add galectin 1, it's galectin 1A in this case, we get extra structures, but if we knock down galectin 1, we get fewer structures. So they have opposing phenotypic effects. And we found that they have reciprocal effects on each other. So CG1A addition phenotype is opposite to the CG1A inhibition phenotype, but it's similar to the CG8 inhibition phenotype, which in turn is opposite to the CG8 addition phenotype. And we looked at numbers of condensations, amount of intercondensation spacing in all these cases. And all of these effects were reciprocal. Okay. But we also found that galectin 1A and galectin 8 activate each other's gene expression, despite having opposite effects on skeletogenesis. So what does that mean? So we're looking here at cells treated with galectin 8, and we're looking at expression of galectin 1A, and we get much more galectin 1A when we treat the cells with galectin 8. Here, if we treat the cells with galectin 1A, we get much more galectin 8. So they activate each other's production. So this is a little paradoxical because they have opposite effects on skeletogenesis, but we found that galectin 1A aggregates limb cells and galectin 8 interferes with it. So we used a turbidimetric method to look at the clumping of cells in the presence of each of these molecule. So if we add galectin 1A, or CG1A is another term for it, the cells undergo aggregation. But if we add CG8, it doesn't cause aggregation. Or if we add a portion of CG8 that is has the combining region for the cell surface ligand, we find that we can inhibit the aggregation induced by CG1A or galectin 1A if we add CG8. And similarly, if we just add it at any step along the way, we can knock down the aggregation. Now, another thing that I should point out is that we also, you could refer to the paper, I'm not going to go into all the details, but we could also look at the effect of these galectins on the cell surface binding component. These are poorly characterized. The only thing that's known about them is that they bind the galectins, and you can assay for them by having labeled galectins and asking whether the cells pick up the labeled galectins. And we found that CG1A induced its own cell surface ligand, which tended to make it stick to the cells more avidly because it was inducing its own binding modality. CG8 bound the cells very well, but it didn't specifically induce its own binding. Okay, so the summary of these experiments can be put in a schematic like this. So this is mesenchymal cell interactions mediated by galectin 1A, galectin 8, and their glycan ligands. And this constitutes a pattern-forming network. And just based on the experiments, we came up with a kind of a very rough idea about what's going on. So the initial condition is that galectin 8 ligand is uniformly distributed. So if there's a fluctuation that leads to locally elevated galectin 1A expression, what it does is that it induces its own ligand, which restricts its diffusion. But it also induces the synthesis of galectin 8, and galectin 8 induces the synthesis of galectin 1A, as shown by the experiments that I described before. Galectin 8 is not particularly confined to the place that it's produced, because its ligand is uniformly distributed and it doesn't induce its own ligand. So it diffuses from its site of production, laterally blocking the activity of galectin 1A. Remember, it interferes with the ability of galectin 1A to cause the cells to clump. But then galectin 1A becomes elevated and active at sites distant from the original one. So you basically get a pattern forming by this dynamics. Okay. But just that kind of hand-waving argument is not sufficient to prove that this really goes on. What we did with our colleague Tillman Glim is to construct a mathematical model that contained all of the experimental components, all of the terms of interaction that we had experimental data for, and terms that we knew existed that we eventually found experimental parameters for. So this is the paper modeling the morphodynamic galectin patterning network in the developing avian limb skeleton. So what does morphodynamic mean? In a standard Turing mechanism, you get a chemical pattern, and then the cells undergo a behavior, a differentiation, for example, based on the chemical pattern that forms. And you don't need the cells to move in order to get the pattern to form. But the cells might move in response to that pattern. So you have a pattern forming, and then the cell executes some activity. We call that morphostatic because there's no movement that's intrinsic to the patterning mechanism. And here is the set of integral differential equations that we came up with that accounted for all of the phenomena that we had observed, or at least represented it. So we have a term, so this is the dynamic of cell density to R. And we have cells being able to diffuse or move, and cells undergo cell-cell adhesion. Okay, and there's binding and unbinding of lectins to their ligands or counter receptors, and there's the change in the density of the counter receptors in response to the galectin. Then there's the galectin 1A, and it involves the fusion of galectin 1A itself, and it involves the response to galectin 8, the concentration of galectin 8, and then there are terms that represent the degradation of the galectins, and there's a similar equation for the dynamics of galectin 8 that involves galectin 1A, everything. Okay, and what we found is that the system could form patterns, and it would form very regular patterns. But take a look at this term over here. This is a cell-cell adhesion term. It's a term that represents the movement of cells in response to galectin 1A, when galectin 1A reaches a certain threshold concentration. And there is a parameter that is alpha sub k that is part of this is part of this term, and that parameter has to be above a certain level. If that parameter is just 200, and that parameter is a component of this constant k here. And if it's 200, you don't get patterns at all. If it's 250 or above, you get patterns, and the quality of the patterns is slightly changed by an increase in that parameter. So there's a dependence of patterning on the adhesion term. This is something that you don't see in the standard Turing reaction diffusion mechanism. So we called this system a reaction diffusion adhesion mechanism to indicate that adhesion is an inherent part of the patterning process. If you don't have a certain amount of adhesion or movement of the cells up the galectin gradient, you don't get a pattern at all. So this system is morphodynamic and not morphostatic. Okay, so then we decided, since patterns of different vertebrate fins and limbs are quite different from each other, we decided to trace back the galectins. The data was becoming available for different species, for different fish, for cartilaginous fish, for ray-finned fish, for the vertebrate organisms with the kinds of limbs that we have, the proximal distally developing limbs. This is moving by itself now. And so we, with our colleague Mahul Chakraborty and and Dr. Sarah Mian, we looked at structural divergence of the galectin 1s. And we found, as I mentioned before, chickens, and this is a zebra finch, they have a galectin that we call galectin 1B. And we know that that galectin does not induce cell aggregation. It does not induce skeletogenesis, and it does not track the formation of the cartilage elements or the pre-cartilage condensations. So this is a non-skeletogenic galectin. So this is the paper that describes the phylogeny of galectin 1, which is a skeletogenic galectin. Okay, did it move?
Yes.
Okay. Here are galectin 1Bs in sauropsids, in birds and in a turtle. Here's galectin 1 in a mouse. And here are 3 different galectin 1s in xenophus. Okay, and just to summarize, I'll tell you that we know that this is not skeletogenic. If we dump this onto cells, it doesn't make new condensations. It doesn't, and this particular protein does not track condensation formation, pre-cartilage condensation. But if we look at the three-dimensional structure of the galectin 1 in the counterpart position in mouse, this is skeletogenic according to the structure of the combining site of the galectin. It resembles galectin 1A, which I told you before was skeletogenic. And among these xenophus galectin 1s, one of them is skeletogenic and the other 2 are not, but they're all clustered here. Okay, did the slide move?
Yes.
Okay. So now we're at a different chromosome. And in the case of the sauropsids, here's again chicken, zebra finch, turtle. We have galectin 1As on a completely new chromosome. You can see that the environment, the contigs are similar, and there's nothing in mammals or amphibians or in fish in that region. So basically, there's been a duplication and a jump to a new chromosome, and these are the skeletogenic galectins by analysis of the three-dimensional combining structure of the galectins. So the galectin 1A gene is on a different chromosome from the other galectin 1s. Okay, and the conclusion of this analysis, and I'm only showing you a little bit of it because it's quite complicated, but the galectin 1A, do you see a word slide now?
Yes.
Okay, galectin 1 gene underwent duplications and divergent evolution in the amphibians. One of the galectin 1 genes translocated to a new locus at the origin of the sauropsids. The sauropsids are the birds and reptiles. Its protein product, galectin 1A, retained the 3D structure found in at least one galectin 1 of all vertebrate species and is skeletogenic in birds. The gene that remained at the ancestral locus in the sauropsids, galectin 1B, evolved to specify a non-skeletogenic protein, galectin 1B. Mammals have a single galectin 1 gene. There was no duplication or translocation at the ancestral locus, but its protein structure retains the presumed skeletogenic 3D structure. So what I'm saying is that all of the vertebrates have at least one skeletogenic galectin 1, but in the sauropsids, it's on a different chromosome, than it is in all of the other vertebrates. Okay, now let's think about the mammals. So we know that mammals have a skeletogenic, or presumed skeletogenic galectin-1. And here is the expression. This is data that's shared with our colleagues Tom Stewart and Gunter Wagner. And here we have galectin-1 being expressed at high levels in the chicken wing and in the chicken leg. And here's the rabbit forelimb and the rabbit hind limb, and galectin-1 is expressed at high levels during limb development as well. But we also found out around this time that if you knock out galectin-1 in the mouse, the limbs develop normally. Is there something different about the mouse? We find that in the mouse forelimb and hind limb, not only is galectin-1 upregulated, but galectin-7, which is a very similar protein to galectin-1, is also upregulated. So there may be compensation and developmental system drift that allows galectin-7 to take over when galectin-1 is not present. But that's the case in mice, but not the case in rabbits. In rabbits, it's very much the same situation that we have in birds. Okay, in any case, so that's one component. And the bottom line is that this skeletogenic protein, the one that induces the condensations, is present in all of the organisms that have a limb skeleton. Okay, we look now at galectin-8, a tandem-repeat galectin, and we looked at the phylogenetic history of that. And we found that in cartilaginous fish, there's a galectin-8, and as there is in all, this is ray-finned fish, the common fish, which has an endoskeleton in its fin, a cartilaginous-based skeleton, in addition to having dermal rays, a different kind of skeleton in its fins. And the Sarcopterigi, which are the lobe-finned fish, which are now considered to include all of the tetrapods, animals like mice and rabbits and ourselves, they all have galectin-8s. And it turns out that the galectin-8 in cartilaginous fish is in the same environment, in the same chromosomal environment, or very similar as the galectin-8s in the tetrapods and the lobe-finned fish. Okay. In the case of the ray-finned fish, there's been a jump of galectin-8 to a different chromosome. And that other chromosome doesn't have a galectin-8 in tetrapods or in cartilaginous fish. So there was a jump of galectin-8. And remember, galectin-8 is the inhibitory galectin. It's the one that interacts with galectin-1 to restrict its spread. Now, in addition, if we just consider these lobe-finned fish, the lobe-finned fish have acquired a conserved non-coding module that is not present in cartilaginous or ray-finned fish. And it's very, very highly marked in all of the tetrapods and in coelacanth, but it's not present in any of the you can look at these numbers. These are high numbers and these are very low numbers, indicating that it's not detectable here and it's highly detectable here. And the non-coding module contains binding sites from mice1, a transcription factor, several other transcription factors. All are mesenchymally expressed during limb development, and mice-1 and runx-2 regulate proximal distal patterning. So this putative control element in front of galectin-8 is a novelty of organisms like us that have true limbs. The fins of the galectin-8 in the fins or anywhere in cartilaginous fish or in ray-finned fish do not have this galectin, this putative regulatory region. So now the galectin-8 is brought under control just in organisms that have true limbs. Okay, so conclusion of this is the galectin-8 gene, which arose in basal chordates, underwent a translocation around the origin of the ray-finned fishes. Galectin-8 protein, now here we apply these computational methods to the predicted protein structure of these proteins. And we found that the ones in cartilaginous and lobe-finned fish evolved 3D structures that were similar to that of skeletogenic galectin-1. And remember, these proteins only take their role in this reaction diffusion adhesion mechanism, only take their role if they are able to compete with galectin-1 for its binding site on the cells so that they can inhibit the galectin-1's ability to clump or aggregate the cells. Ray-finned fish, galectin-8s, generally did not evolve to compete with their skeletogenic galectin-1 homologs. Okay, so the galectin-8 that jumped to the other chromosome did not evolve in the same way as the galectin-8s that stayed behind in the original site. The galectin-8 genes of lobe-finned fishes, including tetrapods, acquired a novel conserved non-coding module that enabled them to be quantitatively regulated in developing appendages. Okay, so just to reiterate, in sharks, cartilaginous fish, and in tetrapods, the galectin-8 stayed on the original chromosome and developed at the protein level the ability to compete with galectin-1A or the skeletogenic galectin-1, whereas the ones that jumped to a new chromosome kind of went their own way. They underwent apparently positive selection that was different in different species. Okay, so let's revisit this again. So the reaction diffusion adhesion mechanism for condensation pattern formation, we have the cell-cell adhesion term, and we have the and there is in this term there is a parameter the beta that is involved in the binding and the unbinding. Here we have a term that has a parameter mu that is involved in the ability of galectin-8 to be regulated quantitatively. Okay. Now we look at this heat map, and here is the galectin-8 expression rate, mu, and here's the galectin-8 binding affinity, beta. Here are the kinds of patterns that form. And you can see that in most places on this two-dimensional space, we get either 0 or very few elements. But if we want to get a lot of elements, you have to be under the curve here. And there is a kind of sweet spot over here where you get the largest number of elements if you have intermediate binding affinity of galectin-8. You don't want it to permanently bind to its receptor because then galectin-1 can't aggregate the cells. But you also don't want it to bind too weakly to its receptor, because if so, it can't compete with galectin-8. And then here, if you modulate the quantitative expression of galectin-8, you can go from a small number to a large number of elements. Okay, this is the number of elements here. And the lower amount of expression you have, the higher the number of elements you get. Okay, so how can we relate that to the different kinds of organisms? So let's look at the conclusion here. For galectin-8 proteins of intermediate binding affinity to their shared receptor, specified by mu, the entire numerical range of parallel skeletal elements is possible. Different levels of galectin-8 expression, which is governed by beta, lead to different numbers of skeletal elements. Only in sarcopterygians, the lobe-finned fish, including tetrapods, has there evolved appropriate mu values and control of beta. And then remember, beta is controlled by that non-coding, conserved non-coding module that has those binding sites for transcription factors to enable proximal distal increase in element number during development. So if we look at the different kinds of finned and limbed vertebrates, we have the cartilaginous fish, which have a lot of parallel elements, very many parallel elements, and some plates that can be interpreted, if you look at their development, as many, many elements that have just fused together. And if we go back here, where in a region of the state space that has many elements, it's up here, okay? And it's this part of the curve where you have these colors, okay? In the case of actinopterygians, the ray-finned fish, you have a whole range of different kinds of morphology. And remember, I mentioned that in these organisms, galectin-8 has not, in general, acquired the shape that allows it to compete adequately with galectin-1 so as to form regular patterns. So you have a kind of non-standard patterns. It's not the same in every fish. You have some repeats, you have some regions that are not repeating, and so on. In the case of the sarcopterygians, you have this is non-tetrapods like coelacanth, and here are tetrapods. This is a shark, this is a human, this is a chicken. You have increasing numbers of elements as you move away from the body. Okay, you don't have just a kind of plate and lots of elements, but you have an increase. It's in the case of fossil non-tetrapod sarcopterygians, you see some indication that this is happening. And in all of the extant sarcopterygians, you see one element followed by two elements, or in most cases of the land ones. And we suggest that this corresponds to the ability to control the amount of galectin-8. So you get well-organized repeating patterns when you have a galectin-8 that can compete with galectin-1A, but only when you have quantitative regulation during development which is afforded by the non-coding transcription factor binding domain, which is a novelty in all of these forms, do you find that you can control the proximal distal progression? Okay, now I'm going to wind up in a few minutes, but I'll point out that there's ectoderm in the limb bud, and there's something called the apical ectodermal ridge, and this is a source of FGF8, fibroblast growth factor 8. And when we look back at the expression of galectins, and we add fibroblast growth factor 8, which is produced by the tip of the limb, the ectodermal tip, we suppress both galectin 1A expression and galectin 8 expression. So basically, as the limb grows out, at the very tip, there is a suppression of just those components that are involved in pattern formation. And the tissue has to move away from the tip in order to undergo pattern formation. And here is a simulation that's based on a reaction diffusion system in which there is production of a suppressing component at the very tip. So let's look at what happens. Okay, so as the tissue is moving away from the suppressive region, it forms condensation. Okay, but the width of that suppressive region changes, and you get transitions to larger and larger numbers of elements as you move out. So you can actually, based on a reaction diffusion dynamics that is suppressed at the tip so that it doesn't differentiate all at once. And if you just control the ratio of the activator and inhibitor and change the shape of the domain, you can actually generate a limb-like structure. Okay. Now, at the same time as this galectin model was being formulated and experimentally tested, there was another model called the BMP SOX9 Wnt model, the BSW model by James Sharp and his colleagues. They had some experimental evidence for it as well, plus some simulations that these components were interacting with each other. Now, SOX9 is a transcription factor that is essential for chondrogenesis. BMP is a morphogen, as is Wnt. None of these components cause cells to aggregate. So our view is that this is a kind of a cooperating system that encourages the mesenchyme to differentiate into cartilage once the cells are permitted to aggregate. So we suggest that this network works in coordination with the galectin network. And then if we look at the evolution, because we know when these different components kind of came into existence in the history of the limbed and finned organisms, here we have skeletogenic galectin-1, BMP, and SOX9. They're all active here. Okay. At 2, we have competing galectin-8 and galectin-1 and galectin-8 cross-regulation. And this enables the formation of the characteristic pattern of the cartilaginous fish, because we can get many parallel elements from the dynamics of the system that's defined by number 2. But then we get to number 3, we get galectin-8 jumping to a different chromosome, and we get positive selection on it. It's no longer competing with galectin-1, and we get variations in the endoskeleton, in the cartilage-based skeleton of these ray-finned fish. It's not stereotypical in the sense that this is, or the Sarcopterygians are. At 4, we get purifying selection on galectin-8, making it increasingly competitive with galectin 1A. And we get the galectin-8 conserved non-coding module. And then this allows us to have proximal-distal development in the limbs. In coelacanth, this is Tiktaalik, a fossil form that was the first tetrapod type of limb that's been identified. It has this format in birds, in amphibians and reptiles, and in mammals. Okay, so basically this is the state of where we are now. We think that we know the relation between the evolution of these galectin components and ancillary components like SOX9, Wnt, and BMP, how they cooperate together to form these cooperating networks to give us skeletal structures. And then we have the regulation of galectin-8, which allows proximal distal development of the limb, particularly when the tip is being suppressed by the fibroblast growth factor produced by the apical ectodermal ridge. So I'll just end there and thank my collaborators. And Ram Bhat was more than a collaborator on this. He basically initiated large parts of this whole project. Tilman Glim has done the mathematical modeling in collaboration with us, and other colleagues of ours, Hans-Joachim Gabi has supplied us with all of the galectin-related reagents that we used in our experiments, and other collaborators were involved in the computational and anatomical analysis. And I'll stop there.
Stuart, I have a question.
Yes.
The BMP SOX Wnt model that you described later,
yes.
Somehow you said it was probably cooperating or acting together with the one that Ramra and you proposed.
Right.
But it seems to me that it could also be thought of as a redundant add-on. Could you comment on that?
Sure. So first of all, unlike the galectin mechanism, which we find is involved at all positions in the limb, not only in the digits, but in the more proximal regions, it organizes the condensations. The BSW mechanism doesn't seem to be as active in the more proximal regions. It seems to be a digit-related module. So it may reinforce the digits, but it doesn't seem to be active in the proximal regions. Similarly, the BSW mechanism has been detected in shark embryos, but again, it's just involved in a series of nodules that are at the very tip of the limb. And if you go deeper into the more proximal regions of the limb, even the cartilaginous rays that are attached to those nodules, that mechanism doesn't seem to be as well regulated in that domain. So we think that the BSW mechanism was acquired kind of coordinately with the galectin mechanism, but was mainly involved in distal organization. And interestingly, since you need SOX9 to form cartilage, and there's evidence that the dermal rays in the ray-finned fish may be also coordinated by this galectin mechanism. You have this option of whether you're going to form what's called dermal bone, or whether you're going to form cartilaginous bone that's templated from cartilage. And the SOX9 seems to be involved in the cartilaginous choice, whereas RUNX2 seems to be involved in the direct differentiation of bone. So that BSW network may be a kind of a differentiation promoting network rather than an intrinsically pattern forming network.
Mike.
Stuart, could you stop sharing your screen so we can see you?
Oh, okay.
So my question is, what about polydactyly? So what happens in individuals with polydactyly?
Yes, let me just see if I can get this back up again. Sorry about this. Am I visible at all to you?
Maybe we can carry on nonetheless. Yeah.
Okay.
The question.
Sorry about that. Yes. So in a Turing type of mechanism, the number of elements that you get, as I showed with the shark and with mainly the shark, you see all those many elements in parallel. It depends on the amount of tissue that you have to organize. So you have a repeating pattern, and the repeating pattern, the distance between the elements tends to be constant and similar to the width of the elements. So if you have a big, broad fin, you get many, many parallel elements. Whereas if you have a narrower piece of tissue, you get fewer elements. And in the case of let's look at the human or the mouse limb, there's an expansion at the tip. So you have 5 digits that form. But at the proximal regions, you have one humerus called the stylopod and 2 intermediate elements called the zygopod. So the number of elements that you get is dependent on the amount of tissue. So in some mutations, like something called TALPID or the GLI3 mutation in mouse, you have a big broadening of the digital plate, and you get many elements. So in the GLI3 knockout of mouse, GLI3 is a suppressor of sonic hedgehog, and it causes a great broadening of the limb bud, and you can have 10 digits, up to 10 digits in the GLI3 mutation. In the chicken TALPID 3 mutation, you can also get 8 to 10 digits because you get a great expansion of the limb bud. But there's also another aspect that addresses your question. It was shown by Sharp's group that the HOX A11 to 13 and D11 to 13 transcription factors modulate the wavelength of the Turing mechanism. So you can actually get thinner or thicker elements and in the same amount of tissue. So you can get fewer or greater numbers of elements in a fixed amount of tissue if you have mutations in these HOX proteins.
Is it on? Okay. In your differential equations that allow the cells to aggregate, how
Yes.
do you, like, I saw that there was a change in density over time, but like,
Yes.
then how do you, like, relative to what? Like, if the cells are stationary, I understand how things would move, but if the cells are moving, like, how do you position them? Does that make sense?
Right. So, yeah, it does. Yeah. So the cells are initially not touching each other. They're embedded in a gel-like matrix, and as you start the dynamics, you get foci of elevated Galectin-1, and those foci attract the cells. The cells move up the gradient of the Galectin-1 so that in any given area or volume, there is a greater number of cells than there is in the surrounding region. And then the cells are then producing more Galectin-1, and the Galectin-1 is causing the cells to produce Galectin-1 receptor. So you get a kind of a auto-regulation of enhanced condensation in those regions. And then correspondingly, you're getting Galectin-8 produced, which drifts away from those sites of condensation and prevents the Galectin-1 from acting on the cells when it's at high enough levels until you get far enough from those peaks of Galectin-8, and then you can start up the Galectin-1 foci again. But there's a kind of a self-enhancing aspect to the movement of the cells.
I got that. I meant, how do you parameterize it in the differential in the ODE model? Like, how do you just have, like,
okay.
In any given area you divide up the space and then the densities change or something like this?
So how is it actually implemented? So yeah, it's there's a module that was developed by I think the paper is Armstrong, and it's basically the cells locomote up a gradient. So basically there is if any point is occupied by a cell, and the cell has an area, the representation of cell material at any point in the grid will change over time, depending on the density of the Galectin-1. That's the best I can explain it, but it's an integral term.
If there are no more questions, I would like to thank Dr. Newman for the wonderful talk on behalf of everyone here. Thank you.
Thank you.